The overall goal of the following experiment is to reconstitute in vitro light harvesting pigment protein complexes of plants or algae. This is achieved by extracting the pigments from spinach leaves or algae cultures and overexpressing the apoprotein in E. coli. As a second step, the apoprotein is mixed with the pigments to obtain a reconstituted complex. Next, the reconstituted pigment protein complex is purified from the excess pigments and the unfolded protein. Results are obtained that show similar spectroscopic characteristics of the reconstituted pigment protein complex and the native one based on absorption spectra, fluorescence spectra, and circular dichroism.
The main advantage of this procedure is that it allows us to obtain a relatively high amount of pigment protein complexes and also to manipulate the complexes using mutated apoprotein and pigment mixtures with different compositions. Demonstrating the procedure will be Alberto Natali, a graduate student in the lab.
To begin the procedure for extracting total pigment from spinach leaves, use a blender to homogenize a handful of spinach leaves, approximately 20 grams, in 100 milliliters of cold grinding buffer for 20 seconds. Filter the solution through 2 layers of nylon cloth with a pore diameter of 20 micrometers and centrifuge the filtrate at 1,500 Gs for 10 minutes at 4 degrees Celsius. Remove the supernatant and add